Hi, my name is Tristan Kriekels, and I'm studying the genetic effects of opioid withdrawal-induced anhedonia using intracranial self-stimulation. So, the big problem in the United States is that 130 Americans die every day from opioid overdose. And what we're doing to research this is we're looking into what makes one individual more likely to become addicted to opioids than another individual. And how we're doing that is we're looking a lot at how different individuals would have stronger or weaker effects of withdrawal the next day after taking the opioid. So, something important to understand is withdrawal-induced anhedonia. Anhedonia is just the inability to feel pleasure, and withdrawal-induced anhedonia is when that is due to the state of withdrawal. So, previous research in our lab has found that higher levels of this anhedonia is protective from further drug use. And in the figures from these previous papers, we can see that on the x-axis, as these levels of WIA increase, the amount of drug that the rats self-administer decreases. And moving forward, we're using rats, of course, and the specific model is intracranial self-stimulation. So the first stage of this is the rats get implanted with an electrode in their medial forebrain bundle. And then when they're in this operant chamber, they're connected to the stimulator. Instead of a lever, we actually have a wheel. But basically, when they spin the wheel, they get an electrical impulse to their medial forebrain bundle, which gives them this sense of pleasure. So we start them off at the highest electrical impulse, and then we slowly decrease it. And if they keep responding and keep responding, we keep decreasing it. And the point at which the electrical stimulation isn't big enough to elicit a response, that's their ICSS threshold. So in our little flow chart, we have ICSS acquisition. That's without drugs or anything. It's just them getting used to the system and getting their threshold stable. And the next stage is spontaneous withdrawal, which is 2 weeks, and we give them the drug. So normally, if a rat were to get a drug and then go straight into the operant chamber, we'd see that their ICSS thresholds would decrease because while on the drug, almost any electrical stimulation would be enough to make them feel good. However, how we're testing them in this research is we give them the drug right after each session so that the next day when they're tested, they're in the state of withdrawal. So what we expect is that while they're in this state of withdrawal, their thresholds actually increase because it takes more electrical stimulation for them to feel good. So, like I said, they do this for 2 weeks, and then afterwards they're euthanized and we dissect their medial prefrontal cortex. So my main research question is, will the anhedonia determined by ICSS thresholds correlate to a difference in gene expression in the medial prefrontal cortex? So here are my first results, and we can see, as expected, the group of rats who received morphine have higher ICSS thresholds than the rats who received saline. And within each of these distributions, you can see that some have higher thresholds and some have lower thresholds. And if we remember from the previous paper, those with the higher thresholds are more resilient to addiction because they'll feel worse the next day and not really want to do the drug again. And those with the lower thresholds will feel closer to normal and are more likely to do the drug again. So, how we relate that to the gene expression side is that we can see our two different groups. We have our high, or the more resilient group, compared to the saline, and the low or more vulnerable group compared to the saline. And DEGs just means differentially expressed genes, So that's adding the up and down regulation of specific genes. And in typical research, more activity in the brain would correlate with more of these DEGs. And usually, when a drug is injected, more effects, more addiction would be shown by more DEGs. However, our research shows that the resilient group actually had more DEGs, which shows that the brain is firing up to protect itself from addiction and it's becoming more epigenetically active. Um, and we saw that throughout with the higher WIA and the ICSS model, which means more resilience, and we found that that leads to more changes in the brain. So thanks for listening. I hope you learned something cool and have a good one.